Breaking the wall of antimalarial discovery. Khoi Nguyen, Falling Walls Lab, Australia.
Think of a hydra, a serpent that can grow back 2 stronger heads for each one that's being cut down by Hercules. Humanity is facing against a similar monster, malaria, a parasite that exponentially targets and destroys our red blood cell. Although there are many drugs on the market used to treat malaria, just like the hydra, malaria parasite has become resistant to all available anti-malarial. Every year, malaria claims more than 500,000 lives, where most of the mortality and cases are in resource-poor settings, where patients can't easily access the treatment that they need. Over the last decade, global malaria elimination progress has stagnated and is projected to worsen with recent funding cuts to research and development. We are in dire need for novel anti-malarial. However, making a drug from scratch is a long journey that can cost up to billions of dollars, up to 20 years of work, and also not all preclinical data can be translated into clinical settings. Most drug candidates fail at human safety and toxicology testing, leading up to 90% of drug candidates fail to reach clinical trials, wasting precious time and resources. But despite the towering challenge of the drug discovery process, there are still 20,000 other regulatory approved human medicine for other indications available on the market. What if they can also be anti-malarial? Sounds simple, but by repurposing existing human medicine for which the human safety profile and dosage has been established, we are only focusing on the medicine's efficacy on the disease of interest, cutting down 5 to 10 years and reducing the cost by up to 90%. Compared to the traditional pipeline. And that's exactly what I've been doing in my master research project in the past 18 months. By studying 600 FDA-approved medicine for the activity against Plasmodium falciparum malaria, or the deadliest species of malaria, I have found 3 candidates that displayed remarkable activity against all stages of malaria that lives in the red blood cell. And upon further testing, I am so pleased to tell you that our silver bullet, which is an existing oral antiviral, is not only available for manufacturing en masse, but it's also so powerful that it has strength rivals some of the current anti-malarial, but the parasite can also not resist it after 6 months of drug challenge. With these exciting results, my lab and our collaborator are now setting up mouse malaria model to test these compounds in living organisms before moving them into human clinical trials. With hope, we would like to bring this lead compound into the market with only 5 to 7 years from now. So fellow innovators, just by repurposing existing human medicine, we are breaking down the seemingly indomitable wall of anti-malarial drug discovery process. By streamlining the drug development process, we are allowing life-saving treatment to reach the 300 million people who are so deeply impacted by malaria quicker, cheaper, and more efficient than ever. Thank you.